Welcome to this explainer. Today, we're diving straight into the elegant architecture of eukaryotic DNA packaging. We are going to unpack the fundamental models of chromatin, the nucleosome, and figure out how exactly the cell pulls this off. To kick things off, let's just consider the sheer, massive physical problem at hand. How exactly does a single human cell fit roughly 2 meters of linear DNA into a nucleus that is, only 6 micrometers wide? I mean, to put that into perspective, it's a structural paradox that demands absolute precision. You can't just shove it in there, right? It has to be perfectly organized so the cell can actually still read the genetic instructions.
Think of it kind of like trying to store a profoundly long garden hose in a tiny shed. It is so much easier if you carefully coil it up. But, coiling a stiff hose takes physical work. In the microscopic realm of the nucleus, this coiling process requires energy. And this energy is cleverly provided by strong electrostatic interactions between the DNA and specialized proteins. These interactions efficiently fold the DNA, which allows it to be packaged into just a tiny fraction of its original volume.
So here's our roadmap for today. First, we'll outline the DNA packaging problem. Then we'll meet the nucleosome, examine the famous beads on a string, build the 30-nanometer fiber, explore higher-order chromatin loops, and finally, decode the histone code.
Section 1. The DNA packaging problem, a spatial engineering marvel. Let's look at why naked DNA simply can't exist on its own inside the eukaryotic cell. You see, packaging DNA into a dynamic protein-DNA complex, which we call chromatin, serves several absolutely crucial functions. Yes, it compacts the DNA to fit inside the cell, for sure. But completely naked DNA molecules are relatively unstable and really prone to damage. So this packaging acts as a sort of protective molecular shield. On top of that, only when DNA is neatly packaged into a chromosome can it be transmitted efficiently to both daughter cells when a cell divides. And perhaps most importantly for day-to-day cellular life, this organization strictly regulates which genes are physically accessible to be read and transcribed.
Moving on to Section 2. Meet the nucleosome, the fundamental unit. This is where the magic really starts. When we look closely at high-resolution crystal structures of this complex, you can see exactly how the DNA double helix tightly winds around a core of specialized structural proteins called histones. This structure is the very foundation of chromatin architecture. And this modular building block is beautifully engineered. As you might know, the DNA of phosphate backbone carries a negative charge, so these histones act as the perfect molecular spool. They use their positively charged basic amino acids, like lysine and arginine, to tightly grip the DNA. Around this octamer spool of 8 core histones, exactly 147 base pairs of DNA are wrapped about 1.67 times in left-handed superhelical turns.
Next up, Section 3, the beads on a string, the 10-nanometer fiber. If you actually look at an electron micrograph of partially unfolded chromatin, you can clearly see this beads on a string formation. Each of those dark little clusters is an individual nucleosome particle. Between each of these nucleosome beads is a stretch of what we call linker DNA. This linker physically connects the nucleosomes together, averaging between 20 to 80 base pairs in length, depending on the species or the specific tissue. By just forming this basic beads on a string structure, the cell achieves its very first level of compaction, shrinking the DNA's linear length by about 6-fold. All right, Section 4, building the 30-nanometer fiber, scaling up. A 6-fold compaction is an impressive start, but to solve our 2-meter problem, the cell has to coil these beads even tighter. To do this, it brings in an additional linker histone, known as H1. Think of H1 kind of like a piece of tape, securing the DNA as it enters and exits the nucleosome spool. With the help of H1, that string of beads folds and twists into a thicker, much denser structure known as the 30-nanometer fiber. So, what exactly is the structure of this fiber? Well, in the widely proposed solenoid model, these individual nucleosome disks stack into a superhelix, featuring about 6 nucleosomes per turn. By adopting this structure, the cell achieves an impressive 40-fold reduction in length. But here's the critical trade-off. Wrapping it this tightly heavily masks the DNA. The flat surfaces of the nucleosomes are buried, which makes the genetic sequence incredibly difficult for transcription machinery to actually access. Which brings us to Section 5, higher-order chromatin loops, the scaffolding. How do we get from a 40-fold compaction all the way to the staggering 10,000-fold compaction needed when a cell actually divides? Let's zoom out again. We've got the primary beads on a string and the secondary 30-nanometer fiber. To reach the tertiary level, these 30-nanometer fibers are gathered into massive radial loops, ranging from 40 to 90 kilobases long. These are anchored at their bases to a non-histone protein scaffold, multiplying the compaction factor to about 600 to 700-fold. Finally, those loops are compressed even further during cell division to create the quaternary structure.
Ultimately, this hierarchical folding creates the iconic, highly condensed metaphase chromosome. This level of compaction is absolutely mind-bending. It is literally akin to taking a rope the length of a football field and methodically compressing it down into a tiny package less than half an inch wide. Crazy, right? Finally, Section 6, Regulation and the Histone Code, Dynamic Control. The crucial point here is that this packaging isn't just a static storage box locked away in the attic. No, it is a highly dynamic, rigorously maintained filing system that actively regulates biology. During normal cellular life, the cell constantly toggles its genome between two states. On one side, we have euchromatin. This is relatively decondensed, forming an open structure where the genes are turned on and actively expressed. On the other side is heterochromatin. Here, the DNA is tightly packed away, densely folded, and as a result, those genes are essentially silenced. Because histones block access to DNA, the cell utilizes specialized nucleosome remodeling complexes. These enzymes use energy to actually slide the DNA along the surface of the histone octamer. It moves kind of like an inchworm, dynamically exposing specific DNA sequences only when they desperately need to be read by the cell's machinery, and then quickly covering them back up. Furthermore, remember those histone proteins that make up the core? Well, they have amino-terminal tails that extend outward from the nucleosome, and these tails act like a biological control panel. Through specific enzymes, chemical tags like acetylation, methylation, or phosphorylation are continually added or removed from these tails. This creates what molecular biologists call the histone code. This code actively dictates how tightly the DNA is bound and whether a specific region of chromatin should be opened up for expression or condensed for silencing. Which leaves us to ponder a truly fascinating concept. As researchers continue to decode the intricacies of the histone code, might we find that the true secret to our complex biology lies not just in the linear sequence of our DNA, but in exactly how, when, and where it is dynamically spooled up? It's a spatial engineering marvel that regulates life itself. Thanks for joining me on this explainer.